Well, as I said, first let me thank Koichi and Jay for organizing this meeting. I was assigned the topic when I opened the program, preliminary program, Future Academic Medicine. I said, yeah, you want to hear about this or want to hear more about science? I can tell you in a short few sentences, future academic medicine is strong but challenged. All of you know that science has never been better. But however, I think we're faced with huge medical societal issues, such as COVID-19, climate change, and many others, which cannot simply be addressed by creating new discoveries or the traditional medicine. So I wrote in 2 papers in Lancet about how academic medicine has to change and has to shift more, not only continue the great work of discovery to translation, but more towards population and population health and global health. I think at the end of the talk, the paper, what I emphasize is the importance of social responsibility of medicine, the social contract that we have with the public. If I can kind of sidetrack, make a few comments, if you look what's happening in our society, in our country, the more recent Supreme Court decisions, the federal judge decisions that interfere with medicine, the practice of medicine is now being restricted in many different states, and there's almost no standards anymore, depends what you practice. So my paper has to do with the importance of academic medicine to focus on this issue for the future. That is, not only do we need to do good science, but we need really to be responsible citizens, and we need to train our students focusing on what's in the future, being actively involved with the social issues, and of course, social determinants. That being said, I just want to say that the topic I've chosen today, I thought that, I hope it's good. So CVBE, I think it's been one of the premier conferences, certainly in cardiovascular research. If you look at the last number of meetings, kudos to Jay and his colleagues. There's been tremendous amount of progress and also great interaction in these meetings. So what I'm about to show you in the first slide is a paper that we wrote in 2021, coming out from one of the CVBE meetings about basic translational research in cardiac repair and regeneration. In that paper, the summary is that the state of the art is outstanding. There are now many different ways to approach the issue of cardiac regeneration repair, such as listed here. And of course, my laboratory and many of yours have worked on different aspects of this cardiac repair regeneration. What I want to do today is to focus one area called RNA therapeutics, which I know many of you are well aware of, and we have started doing some work, starting with microRNA. So why RNA therapeutics? Well, if you look at the way we have approached cardiovascular medicine, small molecules and antibody, they only target 0.5% of the entire human genome. And furthermore, most disease targets lack defining active sites for small molecule binding. What's amazing RNA is they're abundant. They are selectively acting on proteins, transcripts, and genes. So it gives you a much broader array of druggable targets, but also RNA sequences are very well defined, depending on what your target is. That makes the design of RNA therapeutics much easier. And finally, there are drugs already approved and several undergoing phase 3 studies. So I think that, no question, if I want to change my talk from future academic medicine, I say future of cardiovascular therapeutics, very much like RNA therapeutics. Now, just a word about the biology of RNA. At the Academy, we are now taking on the topic called RNA-OME, like genome, in which we have a major study ongoing, likely to actually propose that all RNAs, particularly non-coding RNAs, should be sequenced, and that we should look at individual cells and their environment and disease as well. So our hope is that it will be the next round, like human genome sequencing. But here it is. Everyone knows about messenger RNA, which in fact translate, they transcript and translate in major areas of coding of information. But actually 98% of RNA is never translated. It's non-coding, of which there are about over 10,000 different non-coding RNAs in human genome, which I listed some of them down here. They can be divided into two categories, housekeeping and regulatory. Housekeeping are really important and considerably expressed and required for normal function and viability of the cell. On the other hand, the regulatory ones affect expression of other genes at the level of transcription or translation, such as microRNA, RNA interference, long-coding RNA, and others. Now, it is these molecules that are particularly useful for RNA therapeutics because they can target and influence specific areas and influence expression, and hence, therefore, the basis for different physiology and diseases. So I tried to make a list of what I would consider RNA therapeutics. When I read up some papers, they listed antisense RNA as well. Now that brings back memory. When I was at Stanford in the 1990s, we worked a lot on antisense oligonucleotide with some of my students like, Ruichi, Murishita here, and many took all the way to phase 3 studies in some of our work. But also microRNA, RNAi, messenger RNA, and more recently the CRISPR, in which you do prime editing of RNA editing as well, and of course the synthesis of aptamer. I'm going to touch on some of this in my talk. So many of you know that my lab pioneered the whole idea of microRNA in reprogramming of fibroblasts to cardiomyocytes. We have discovered through combinatorial approaches 4 microRNA combination called RNA combo, microRNA combo, microRNA 1, 133, 208, and 499. What you can see on the left-hand side is a heat map of gene expression, first looking at the bottom, which is control, using control, of course, RNA. They're not targeted. But importantly, you can see these are fibroblasts with high levels of expression of fibroblast-mediated genes. But using different mRNA combinations, you can see there's a shift in the expression from fibroblasts to cardiomyocyte or cardiac genes. And we found that in microRNA 1, 133, 206, 208, or 499, you can see in the middle panel, in fact, the cells of fibroblasts became cardiomyocyte-like with staining for actinin and having contractility as well. More importantly, recently, we've shown that it actually works equally effectively in pig and human. As you can see, this is RNA-seq on the right-hand side, where you can see the large number of genes which are being expressed in response to the microRNA combination, and of course, the production of cardiomyocyte, as you can see in the right-hand side. Now, you may or may not know we have entered large animal studies with this micro combination to see whether, in fact, they could be applied to human application. Now, I think many of you have seen this data before. It's a composite of work that we've done where we look at these 4 microRNA combination using mice with lineage tracing for fibroblasts. So FSP1 Cree tomato will allow you to look at any cells derived from fibroblasts to be stained with fluorescent protein tomato, red. Consequently, when you reprogram these cells into cardiomyocytes, you have staining for cardiomyocytes, but in cells that were red, which are derived from fibroblasts. As you can see here, here's a stain of a myocardium after 4 weeks of acute myocardial infarction of mice. And you can see there are islands of cardiomyocytes, which are derived, as you can see, from fibroblasts. So consequently, the microRNA combination works in the sense that we reprogram fibroblasts into cardiomyocytes. Importantly, when we took the cells and studied ex vivo physiologically, see, this is the wild type with no tomato staining. This is now fibroblast-derived cardiomyocytes. The action potential, the calcium oscillation, and contractility are almost identical, so they're physiologically normal. And we also demonstrated that it restores cardiac function as fractional shortening, left ventricular end diastolic volume, and as you can see, there's significant improvement 2 months later. So in this talk, I'd like to focus more on how does microRNA affect reprogramming and eventually the formation of cardiomyocytes. because it would give us insights into how to improve therapy, particularly in RNA therapeutics. Here what you can see is if your fibroblasts were going to differentiate and reprogram into mature cardiomyocytes, there are at least two major steps. One is the initiation, which allows cardiac-specific lineage to be committed to, and second is the maturation. Of course, there are many steps in between. But importantly, when we looked at the microRNA combination mechanisms of action, we found it actually acted both levels of transcriptional control and activating a series of RNA sensing receptors. So what I'm going to show you some data that relates to this. So first, transcriptional control. Our laboratory has published several papers, including one by Sophie Deprau in 2017 in Circ Research, and more recent in JBC, in terms of looking at how the combination of microRNA affects transcriptional control. First of all, microRNA combo will inhibit EZH2, which is an essential protein that's important for the activation of H3K27 methylation. Ergo, it inhibits methylation and causes demethylation, which actually causes an activation of gene expression. At the same time, there are repressor proteins or repressor genes that represses the cardiac gene activation and keeps the fibroblast in fibroblast lineage. And this is shown over here. So consequently, microRNA combo actually inhibits repressor mRNA and therefore activates transcription. So with these two actions, which I'll show some more data, we can explain how microRNA actually, as an RNA therapeutics, influences transcription control. We have published recently the discovery of CBX1, PERB, and SP3 complex as three genes that actually important in silencing of cardiac gene expression and keeps the fibroblast lineage, fibroblast in its own lineage. Here's some data to show you the evidence. So what we did is we look at all the highly expressed genes in fibroblasts which are low expressed in the cardiomyocytes. And taking each one of them, we actually knocked out each one of them and look at what happens to the cardiomyocyte gene expression. You quickly see that three genes, CBX1, PERB2, and SP3, when they are knocked down, if you will, by siRNA, they become cardiomyocytes at high levels of gene expression. Shown over here is the number of sarcomeric actin, which are stained % of cells, where you can actually see that by each one of siRNA, we now have gone from 0 to actually a good 10 to 15% of cells expressing sarcomeric protein. We then went on to do the experiment in vivo by looking at CRISPR and using guide RNA. And first, you can see that we successfully in vivo knocked down CBX1, PERB2, SP3. And over here, you can see control guide RNA and what happens to lineage-traced fibroblast origin when all these things are being knocked down. Here you can see clearly cardiomyocytes originating from fibroblasts by the suppression of these three repressor genes. So I think it's fairly clear that we are dealing with, in fact, repressor genes which are mRNA targets, because when you look at mRNA given to fibroblasts, you can see that there's a suppression of CBX1, SP3, and PERB3. So they're targets of microRNA combos. And importantly, when you look at iPS cells, differential cardiomyocytes, here you can see a significant suppression of these three genes as well. So in other words, these repressors are important in keeping the fibroblast in fibroblast lineage, whereas the removal of these suppressed repressors will activate, and together with the epigenetic effect of the microRNA allows the cells to become cardiac lineage. So in summary, in this part of the work, what we've noticed is cardiospecific genes are actively repressed by repressors, and they mediate repression via the H3K27 methylation deposition. And therefore, microRNA initiates reprogram by reducing the expression of these three, and therefore allowing cardiac gene expression. Now, the second phase of our work looks at what causes maturation? What is the effect of maturation, particularly the work by the microRNA? And this we have done it in collaboration with John Cook, where we have reported that toll-like receptor TLR3 is an important mediator of the microRNA effect in mediating through NF-kappa B maturation of the cardiomyocytes. We published this in Stem Cells a few years ago, and we continue to do this work by demonstrating, first of all, the evidence that TLR3 mediates the mRNA effect on maturation. Here you can see in the heat map the expression of large number of cardiac myocyte genes by microcombo, not seen by negative MAIR. But when you add an inhibitor of TLR3, you can see the expression is greatly downregulated, as shown over here for myosin 6, actin 2, and troponin 3, where you have marked increase with microRNA combination, but that in fact is blocked by the blockade of TLR3. TLR3 is part of a family of toll-like receptors, as shown here. But importantly, there are 4 toll-like receptors, TLR3, 7, 8, and 9, whose agonists are RNA molecules. And they actually belong to a family of RNA sensing receptors. So what you see here is not only these 3, but also RIG1 and MDA5, all of which are sensing RNA, double-stranded RNA, in which they sense virus that can activate innate immunity. So in so doing, we begin to ask the question, what about the other RNA sensors? We have just published this paper in which we demonstrate that RIG1, the other sensor, play a critical role in cardiac reprogramming, and it's done through 2 mechanisms of signaling, NF-kappa B and YY1. Here you see data in terms of TLR3 agonists with microRNA, which greatly increases the amount of expression of cardiac myocyte genes from microRNA combo alone. And this is an agonist of RIG1 agonist, which also does the same thing. Now using NF-kappa B inhibitor, the Bay compound, we can see that it mediates in part through NF-kappa B. But in our paper in JBC, we also demonstrated that it also affects through YY1. As you can see here, MAIR combo increases expression, cardiomyocyte gene expression, but the addition of the agonist for RIG1 results in further increase. But when we actually deleted YY1, you can see it brings it down towards this level. So when we have done more further work in that paper, look at immunocomplex formation, and came to the conclusion, this is how it works. That in fact, MAIR combo activates fibroblast initiation in terms of cardiac lineage gene expression. It further is a weak agonist of TLR3 and also RIG1. That TLR3 RIG1 activates not only NF-kappa B, but also Y1, which form a combo with KDM6 and PO3, which then result in actually demethylation that in fact activates the expression of cardiac myocyte genes. So with that background, it became quite obvious in terms of RNA therapeutics. One area is to do synthetic RNA called aptamers in terms of looking at how to further enhance the effect of the RNA sensing receptors. And the best way, of course, in collaboration with Bruce Selinger, is to aptamer screening. Small RNAs that interact with receptors and see how they activate it. This is already published in the Nucleic Acid in collaboration with Bruce Selinger's lab, where we have synthesized a series of compounds, synthetic RNA agonists, called ICRs. And as you can see, ICR2 and ICR4 also markedly increase the microRNA combo effect, particularly in ICR2, see here. And of course, the poly-IC is the agonist of TLRs, but it's highly inflammatory, but ICR, through its selection, is non-inflammatory. So now we have evidence that we have synthetic oligonucleotide RNAs that can actually activate these RNA sensing receptors. But I think an interesting observation is the recognition of set of ligands of each RNA receptor. For example, TLR3 recognizes long double-stranded RNA molecules. Reg1 recognizes RNA molecules with 5' triphosphate group. So our question becomes, can we actually modify the microRNA through 5' triphosphate so that it becomes 5' triphosphorated microRNA, which will then have a combination of both the Reg1 activity as well as microRNA activity. So the question is, what would modified 5PP have on reprogramming and immune response? So this is a summary of data, which in fact is also impressed. What you can see is RNA-seq on the left-hand side, where you can see that upregulated genes are in yellow. MicroRNA combination results in shift from fibroblasts to cardiomyocytes. And importantly, the neg-mir of 5' has no effect, but 5' microRNA markedly further increases the level of expression of cardiomyocyte genes and downregulation of fibroblast genes. This is PCR data showing you, actually, this is just microRNA, but this, in fact, is the 5' microRNA, which markedly increases. In fact, microRNA 1 5' triphosphate has about 80% of effect of all microRNAs, and that alone, without using 3 microRNA, can do much of the effect by converting it into 5' triphosphate. Here you can see the number of cardiomyocytes per field in vitro, which markedly increased with 5' triphosphate microRNA, and you can see the number of fibroblasts. The number of beating cardiomyocytes markedly increased. Individually, these cells are physiologically normal, and importantly, in fact, the beating oscillations are also towards normal cardiomyocytes. So modification of microRNA combo by 5' triphosphate is a very effective way of increasing microRNA effect. This summarizes some of our findings. MicroRNA itself, combo, can activate NF-kappa B and therefore resulted in expression. Whereas 5' brings together YY1 and complex that further result in demethylation and markedly increase in expression. And this is shown, in fact, over here when you can see that we're shifting from a methylation to demethylation by the combination of 5' modified microRNA. So I've given over a lot of work that we've done in microRNA, but what about the other RNA therapeutics? I've talked about aptamer, microRNA, and 5' microRNA. So I'd like now to shift towards more speculative data about messenger RNA, because all of us know about mRNA platform and what's done in vaccine production. So it's a very effective way of producing, in fact, proteins that can be useful as a biologic or as an antigen for antibody response. I know that Ken Chin was involved with this work some time ago. It's now in AstraZeneca, in which this EpiCure study have now reached 7 patients with microRNA VEGF. Now, while the study is still ongoing, it's primarily safe. So far, the endpoints have not been positive, but it's too early. It's really more of a safety trial. But I think the main thing to point out is mRNA can work as a platform for cardiac regeneration. But it's not VEGF, then what? That brings me back to my favorite, the hypothesis of paracrine. As you know, our lab was the first or the pioneer in paracrine hypothesis. You can see the large number of papers we have published on this area, and more recently, the idea of sequential activation of genes. So if one were to take that as a thinking, previously, our aspiration was to say, if we find those novel factors that actually are paracrine factors, how do we turn that into therapeutics? And that was the challenge, trying to take recombinant protein to inject it into myocardium. But now with mRNA platform, it's certainly possible now to start testing some of these paracrine factors as therapeutics. So this slide shows you, of course, that paracrine hypothesis, which says that it can work through many different paracrine factors in terms of anti-apoptotic, anti-fibrotic, new vascularization, and cardiomyocyte differentiation and proliferation. Hence, all of these become potential mRNA therapeutic targets. That is more defined secretory profile to improve outcomes, to look at novel paracrine factors we have reported and discovered, which includes ABI3BP, HASF, and SFRP2, to look at regulation of immune response, and of course, looking at program efficiency. I'm going to show you data in terms of SFRP2 because I think this is clearly, in my mind, one of the prime candidates for mRNA technology for paracrine release and looking at cardiac regeneration. SFRP2, my lab has studied SFRP2 quite a few years, as you all know, and it's a paracrine factor released initially by initial work on mesenchymal stem cells. It's a modulator of Wnt signaling through direct interaction with Wnt, which I'll show you in a few minutes. And of course, our work and others have shown important roles of SFRP2 in inhibiting fibrosis and promoting myogenesis. So SFRP2 acts on different parts of the regenerative pathway. It can reduce apoptosis in early stage. It can reduce fibrosis, increase angiogenesis, and I'll show you data. It can certainly increase regeneration by activating cardiac progenitors to become cardiomyocytes. First, on the fibrosis remodeling. It's been shown that BMP1 is involved with post-MI fibrosis. Our data and others shown that SFRP2 can directly bind BMP1 and modulates activity, and hence, by injecting it after post-MI, within 2 days, you can see reduction in the fibrosis and collagen deposition. Here you can see a paper published in PNAS, which SFRP2 in vivo, 2 days after injection with after MI, it inhibits collagen maturation fibrosis in vivo, which you can see very clearly over here and here. And importantly, we look at collagen maturation fibrosis, you can see that, in fact, the fibrotic areas greatly decrease in 1 week and 2 weeks. And overall, there's much less fibrosis after SFRP2. We've also demonstrated previously that SFRP2 can induce mesenchymal stem cells to become cardiomyocytes. But more recently, we published that it can actually is a very good inducer of human iPS cells to mature cardiomyocytes. In fact, this paper was a methodology paper which demonstrates that the current way of inducing iPS cells to cardiomyocytes is not as good as using SFRP2. I ask you to look up that paper. But my main point is to say that SFRP2 can induce myogenesis. And the mechanism which we have studied over the years is that first, SFRP2 binds directly Wnt3A, which is a canonical Wnt. And this canonical Wnt will activate beta-catenin and inhibit cardiac specification. But by binding Wnt3A allows Wnt11, which is a non-canonical Wnt, to bind to frizzle receptor, activate junk and calcium activation, which actually causes myocyte differentiation. So that's, in fact, how it works through many of the work that we have done. This is a study published in PNAS, which we looked at with SFRP-induced myogenesis. Well, not knowing what the cardiac progenitor is in the myocardium, as you know, there's a lot of debate what it is. We actually took a chance to look at what are the potential progenitors, one of which we looked at c-KIT. We all know that c-KIT is highly controversial, but nevertheless, we took a look at it and we did, again, lineage-specific tracking of KIT cells. So here you can see KIT cells with Cree with the tamoxifen activation to actually co-express GFP. In other words, if a KIT cell lineage, you should be able to detect it with in green in terms of GFP. And what we did is we injected SFRP2 to after myocardial infarction and look what happened. First, at vehicle after MI, you can see that there's really not a lot of activities here, except for a lot of cardiomyocytes. You can't see that very well, which is stained in the red. But with SFRP, you can see now the green color in cardiomyocytes, which means these are cardiomyocytes originated from kid cells. If you look at baseline after MI, there's a lot of endothelial cells, which kid cells are able to become, as you know, angiogenic endothelial cells. But whereas SFRP switches the kid cells over to cardiomyocytes. And importantly, these myocytes are integrated with connexin 43, and you can see all the ones in vivo are integrated. And when you study them ex vivo, you can see the calcium transients are similar between kid-derived cardiomyocytes and wild-type derived cardiomyocytes. So in other words, during this study, we actually started tackling one of the controversies. It cells a progenitor of cardiomyocytes. The answer is usually not. If you have MI, they activated form angiogenesis. However, under right condition, in this case, such as SFRP2, they can indeed become cardiomyocytes. So this actually is the data that showed that with SFRP, we have increase in ejection fraction, in fraction shortening, reduction in LV systolic and diastolic volume, and ultimately the amount of muscle that's markedly increased through regeneration, reduction, in fact, in scar. So I've given you some idea that, in fact, I believe our next work would be to actually put SRLP in an mRNA platform to see what we do, because now we suddenly have a much easier way to make this work than using recombinant protein. So I want to end by talking about RNAi. So in RNAi, there's already quite a lot of work in clinical trials, and the number of drugs being approved for example, amyloidosis and neuropathy, and in the midst of looking at cardiomyopathy as shown here. So I want to use one example of recent work as target for RNAi therapy. This is in fact looking at neonatal and cardiomyocytes and how they are different in regenerative capacity. In adult cardiomyocytes, the endothelial pass containing domain gene called PAS1 is expressed where it's not, in fact, in the neonatal myocytes. We have to actually ask this question: if one were to knock this one out, would we actually end up with much greater potential for regeneration from fibroblasts to cardiomyocytes? And here's what you see here. By using siRNA in vivo, we've been able to show, maybe that's the next slide, this is in vitro, able to show, in fact, it is inhibiting the effect of the capacity for fibroblasts to become cardiomyocytes. And in vivo, which is published now in JBC, you can see that there's clearly using, in this case, exosomes containing a combination of microRNA and siRNA, we actually inject it in the border zone. In so doing, you can see that, first of all, in the combination of control and microRNA, there in fact is the formation of cardiomyocytes. But by knocking this down, there's even much greater formation. And in vivo, you can see increase in ejection fraction, fraction shortening, reduction in LV systolic diastolic volume, and overall a regeneration of the myocardium. So this is an example in which siRNA or RNAi can be used for cardiac regeneration. So now, I think now that I've given you kind of a tour about how RNA therapeutics can work, let's face it, there are limitations. Even though it has tremendous potential, it has overcome extracellular and intracellular barriers because the large hydrophilic negatively charged properties prevent RNAi from passively crossing lipid bilayers. And also, these drugs has to evade serum nuclease and scavenger macrophages within the RES system, has to cross the cell membrane, escape endosome, and release mRNA, RNA into cytoplasm. But all this can work. The main thing is chemical modification. As you know, Drew Weissman and Kalina Kiriko modified the mRNA to reduce inflammatory response and, in fact, increase stability, and that overcame the whole issue of use of mRNA. So chemical modification of RNA is going to be a key factor in enabling RNA therapeutics, but also delivery. You know, I told you a story back in 1990s when I was at Stanford. We did a lot of work on antisense oligonucleotide. we're kind of pioneering, if you will, on quote gene therapy. One of the biggest limitations was delivery, and delivery remains to be a limitation today. So what we have to do if we target fibroblasts is to figure out how to have target fibroblast-specific delivery, and that's a real challenge. As you know, in the AAV viruses, there are some isotypes which are more fibroblast preference, but not specific. And of course, in exosome, there are exosomes which are more fibroblast specific, but not fully effective. So we're now collaborating with Erivan Elsokan, who worked with Jude Samolsky, who's now a Duke professor, in using the approach of molecular evolution. The idea is to use, as you can see, a capsid library, generate a AAV capsid library with a large number of variants with spontaneous mutation, whereby there are lots of potential fibroblast-specific tropic AAV. Then you go through rounds of selection, through both in vitro as well as in species selection. And you can see we now have very interesting data that one of AAV1 has much more fibroblast specific than the cardiomyocyte, but that's still very much work in progress. Second is to look at exosomes. We have actually reported an C166, an exosome derived from endothelial cells, which are more fibroblast specific. But more recently, we have data, which I'll show you here, in targeting fibroblasts specifically through the addition of fibroblast activating protein antibody targeted towards it, which is single chain FV fragment of an antibody, immunoglobulin, which is targeted towards FA, fibroblast activating protein, shown here. So in so doing, we can show definitely that actually it's more fibroblast targeting in exosomes. So that's obviously very encouraging. But at the end of the day, certainly for mRNA, nanoparticles are going to be critically important, as is for RNAi. I used to be on the board at Alnylam. In fact, I remember the day where, as a board member, we say, let's have a target where we're going to put RNAi. We think about RSV1 and the brain. We came with the idea that, in fact, if we have a nano delivery system that targets the liver, GalNAc, we can treat all the genetic diseases whose gene product comes on from liver. Hence, now you can see large number of successful products. So I think delivery is going to be a major issue for RNA therapeutics. So I want to summarize by saying that certainly we do have large number of possibilities that promise RNA therapeutics. Already I've shown you, certainly in vivo and in vitro, the effectiveness of microRNA and 5' triphosphate microRNA and the aptamer. RNAi has already been shown to be effective in human, but now I've identified some targets for, in fact, cardiac regeneration. mRNA would be a phenomenal technology to look at the parenchyma hypothesis and the various secreted material where you don't have to worry about cell specificity anymore. You just have to deposit and release it as a parenchyma fashion. And of course, I haven't talked about prime editing, which is an emerging area of great importance. AAV, exosomes, and nanoparticles have to be effective agents for delivery. I think that's going to be the big challenge that we all face with, although all of them holds great promise. So I want to end by thanking my laboratory, particularly Conrad Hodgkinson, who's done a lot of that work, along with myself and many others listed here, my collaborator John Cook, Paul Rosenberg, Nanette Bursek, Bruce Salinger, and Erivan Elsokan. Thank you very much.
So stay here, Victor, Dr. Dzau. So I always like to have interaction with the audience. So this is go back 3-4 decades, great science. So we have time for 1 or 2 questions. Anyone? So this is going to be half a century and a new chapter of the cardiovascular medicine, and COVID gave the RNA therapy a new life. So, John?
can't hear you.
How about that?
Yeah.
Thank you for that wonderful overview and also for your leadership over the years of PCTC and PCBC and this consortium. I'm glad you brought up the issue of inflammatory signaling that's induced by microRNA and messenger RNA. Virtually all of the molecular therapies that we're using do activate inflammatory signaling and increase chromatin accessibility, as you showed, that it's very important for reprogramming. And we've given it a name, and I think it's important that that process has a name so people remember it. Transflammation. We should all remember that our molecular approaches can induce inflammatory signaling to have an effect that's important for reprogramming, the chromatin accessibility, as you mentioned. I'd like to hear more about how you're manipulating that pathway with your aptamer approach.
Yeah, so first, as you rightly pointed out, that actually my own feeling is in cardiac reprogramming and where we do the immune mechanism is really important. You're going to hear some more discussion later. To me, it's not the same as inflammatory per se, because as you can see, it can reduce inflammation while activating some of the innate pathways. So I think a lot of work has to be chemical modification. In the aptamer work we did screening to look at what are the ones that optimally have agonistic effect and minimally the inflammatory response, and that's how we do it at this point. But as I said earlier, if you look at the work of Catalina Carrico and Drew Weissman, yeah, they actually modified, in fact, some of the chemicals, RNA nucleotides, in order to reduce inflammation.
Yes, that's true. The modifications do reduce inflammatory signaling, but I think actually the benefit of the RNA vaccines were in part because you still have some inflammatory signaling. It's still activation of the pattern recognition receptors despite those modifications.
Well, I don't doubt there's some effect, but we all know the early work, the reason that they actually got the big prize is because it was not doable initially. It's such a simple concept by putting mRNA into cells, right? But it was all rejected by inflammatory response. So I think, yes, I'm not debating there's no need for immune response. I'm just simply saying that you're going to have to get it right.
It's a great talk. Thank you so much. The human genome contains a number of pseudogenes that most of them are actually cDNAs that are translated or reverse transcribed and inserted into the genome. And they probably have regulatory functions, and I'm surprised nobody is trying to target those because the RNAs of I'm saying that wasn't on the list. Do you think anybody is working on that to increase the expression of some of these pseudogenes for regulatory function?
Not to my knowledge, but I could be wrong. Certainly the project we're taking on at National Academies is to look at sequencing all the smaller RNAs or the cDNAs as well. All right. Thank you.
Great. So
Thank you.
you are going to